Hello students, welcome to the theory class of Principles of Plant Breeding. We will discuss on the breeding of biotic stress resistance. Actually the main topic is breeding for biotic and abiotic stress resistance. Okay, so since it is a large chapter, I have divided this into two parts: breeding for biotic stress and breeding for abiotic stresses, because both these approaches for breeding in biotic stress and abiotic stress is different. The reason why I'll tell you. In case of biotic factors, what are the biotic factors? We can imagine it can be a fungi, it can fungal disease, it can be bacterial disease, it can be any protease or viral disease, or any kind of living organism, okay, which affects the plant, which affects the yield. Okay, ultimately if I say which affect the plant, and if it doesn't affect the yield, it also doesn't come under um, the biotic stress, because nowadays on the plant breeding perspective, okay, even though if the insect feeds the plant and if it yields the same results, or if it yields higher than the infected plant, okay, it will we can still call it as a tolerant plant. Okay, we can still take it up as a good genotype. So we are going to talk about here the biotic factors and the mechanism which are which we can use, or which you can promote to resist against these factors. Okay, so in case of biotic factors, there are other factors which I'm not going to discuss here, such as rodents, okay, such as say hogs and cattle. Okay, these are also biotic factors which affects the uh, croplands, but not in major. And when it comes to biotic factors, the main emphasis and the main biotic factors for crop loss in agriculture and allied sectors such as agriculture and horticulture is our diseases. Again, diseases supposed to mean the biotic factors such as fungal disease, bacterial disease, and viral disease, and the insect, okay, the main, which is infected insect, and of course nematodes are also there. And on the other hand, the abiotic factors are air, okay, lack of what do you call, carbon dioxide in the air, okay, or pollution in the air, okay, and salinity, okay, salinity which decreases the nutrient uptake capacity, soil, acidic soil, sodic soil, okay, or barren land which makes the land barren, okay, and high amount of lead content in the soil, okay, or submergence, flooding, drought, okay, all kinds of, or and at the same time uh, insufficient light intensity, okay. So all kinds of abiotic factors which we cannot manipulate are the abiotic factors, okay. So this is the main uh, the main motive here would be to modify the plants, and we cannot control abiotic factors, okay. Most of the time it is erratic, we cannot have a control. So sustainable use will be the main objective, and at the same time modifying the plant which can sustain the the changing climatic conditions, or you can say weather conditions. And of course it is quite related to the current situations, the current climate challenges, the climate change, okay, much talked about topic of these days. And today we're going to discuss about biotic factors only, as we have already said. And the next slide we have here is the losses which is accounted from both biotic and abiotic stresses. The lines, the bars in orange color is are the losses due to abiotic stresses, okay. And the the bars in blue color is representing the losses due to biotic factors, okay. So most of the biotic factors are due to insects again. Okay, so in this, when we compare the biotic and abiotic factors, the major losses in agriculture and horticulture crops are due to abiotic factors. Next, coming to that, we have biotic factors. Okay, and to start with, we need to clearly have an idea about what is quantitative traits and what is qualitative trait. Okay, I have already mentioned about the the difference between the biotic factors and abiotic factors affecting the crop plants. The approaches are different. Why? Because the genes which controls the biotic factors and abiotic factors are different. That's why we have to have a different approaches for both these kind of stresses. Okay, so what is quantitative stress? Okay, what the quantitative, in case of quantitative stress, as you can see here, it is a complex trait, okay, and which is controlled by many minor genes in cumulative action. Many minor genes in cumulative action, it means it lots of genes are involved where their action is small, minor, but they have a cumulative effect on the major traits such as yield. Okay, so when you have the major cumulative effect, so it is hard to transfer into a new type, okay, new plant type. But on the other hand, the qualitative genes, okay, so in case of qualitative genes, it is discrete type of, or you can say qualitative traits, it is a discrete type of trait. So when in terms of biotic factors, qualitative trait applies, because in biotic factors we have either a disease resistant plant and a disease susceptible plant, or under that if you can put a third one, which is disease moderate disease plant. Okay, so a plant is either disease resistant or disease susceptible, so we can categorize this under two heads, resistant and diseased. Okay, so it is quite clear and it is easy. The genes which controls by controlling these traits are easy to identify and easy to transfer from one individual to another or wild type to another, while in case of quantitative traits it is difficult to transfer from one trait to another because the genes are uh, quite huge in number and they have a cumulative effect. Okay, so all kinds of environmental effects are also contributed in this. So it is a complex process, and when it comes to qualitative treat, is a simpler process. Okay, next is coming to the next slide. We have the mechanisms of insect resistance. There are four mechanisms here: non-preference, antibiosis, tolerance and avoidance. The first three, which is given by Painter in 1951, and the fourth one is followed later on. Okay, so the first three mechanisms of insect resistance is correlated as seen in the diagram, and the non-preference means it is a mechanism in the morphological structure of the plant body. So, for example, hairiness of the plant body, which makes the, the insect non-preferring for oviposition, fooding and sheltering. Okay, and when it comes to antibiosis, when the insect feeds on the plant body, thus the signal is sent into the plant body with signal transduction, and there is a hypersensitive reaction. Okay, and then the the which affects the, which sends uh, phytotoxin into the leaf or feeding area, and which makes the plant, or which makes the insect uh, which affects the insect, okay, which feeds on the plant body. Okay, so some of the examples may be the bt cotton may be a good example of antibiosis. When the bt, when the Helicobacter armigera, popularly known as cotton bollworm, feeds on the plant body, so it sends signals on the uh, plant mechanism, and the delta endotoxin is released. Okay, upon feeding the delta endotoxin, so the insect larvae freezes and dies. Okay, because it works in the alkaline pH. So next is tolerance. Again, tolerance is what? Uh, Tolerance is the mechanism of the plant body which can sustain the infestation of insect. Okay, or which means which can have a normal yield as it was there in resistant plants. Okay, or uninfected plants. When it comes to non-preference, as we have already told, it is also known as anti-zoonosis. The host plant, the host variety that are unattractive or unsuitable for colonization, oviposition, or both for insects. Okay, and the characters adopted by plants include hairiness, sometimes different in color and leaf angle, etc. Okay, so hairiness of leaves making plants difficult for oviposition, feeding, etc. Some of the important non-preference mechanism here are given is in case of wheat we have stem, solid stem instead of hollow stem. Okay, and in case of rice for stem borer it is lignified stem instead of non-lignified stem, and for brown plant hopper low aspergillus content instead of high aspergillus content which is preferred by the insect. Maize, in case of maize, for corn earthworm we have toughness of husk instead of soft husk, and corn leaf aphid, for corn leaf aphid the non-preference mechanism would be containing high dimbo content. Dimbo is a chemical. Okay, and low dimbo content uh, in preference mechanism. Okay, so and so in case of cotton bollworm also. Next one is antibiosis. Okay, antibiosis refers to the adverse effect of host plant on the development of reproduction of insect pest which feeds on the resistant plants. Okay, and in some cases antibiosis may lead to even death of the insect, and it is considered as a true form of resistance for insect pest. Okay, some of the examples would be sugar cane, in case of sugar beet, for aphid, low sugar, low free sugar, for alfalfa, in case of spotted aphid and pea aphid, high saponin content is an antibiosis mechanism. For brassica, in case of brassica against cabbage aphid, low cyanigrine is an antibiosis mechanism. For potato against aphid, gummy trichome exudates is a antibiosis mechanism. And for tobacco against mites, exudates of glandular leaf hairs is again a antibiosis mechanism. Okay, and there are other examples for other crops also. These are a few of the many examples. Okay, so in case of tolerance, okay, the picture depicts about the resistant plant, susceptible plant, and tolerant plant. So in case of tolerant plant, the the yield will be the same as that of the resistant plant. In case of resistant plant, what we can see, you can see is the insects cannot affect on the plant body, but in case of tolerant plant the insect is infecting the plant bodies, but still the yield is the same. It either it is not affecting the fruit, but then if it affects on leaves also, the photosynthetic activity still goes on, and in such a way that the yield is same. Okay, and in case of susceptible variety, it when the insect infests on the plant body, so yield will tremendously decrease because the photosynthetic activity is well decreased. Okay, so this is the mechanism of tolerance. And the last thing and the most important one is avoidance. Okay, what is avoidance? It refers to the escape of variety from insect attack, either due to earliness or due to its cultivation in seasons where insect population is low. For example, early maturing cotton varieties for pink bollworm, and so okay. So in case of this can well suit to the recent example of migratory insects, or what you call it as locust. Okay, so locust is a common migration of locusts from one place to another is a common phenomena, and during summer season in in North India and in African regions. Okay, so in this, especially in this, what farmers used to prefer is farmers used to empty their field plot. Okay, and for this the best thing to do is to harvest the crop before the arrival of these locusts, okay, the migratory or gregarious insects. So once they arrive, they don't leave anything, they feast on everything, and it leads to huge loss in the crop or the farmer, okay. So not only this, in case of disease also, okay, there is example of tika disease, that is what disease. So if you harvest the the groundnut well before the season, okay, well before late rabi season, so you can avoid tika disease in groundnut. And example of one example is also given here: early maturing cotton varieties. If you harvest cotton well advanced pink bollworm, you can avoid bollworm also, okay. So some of the methods for breeding of disease resistance are given here. The first method would be selection, which is the easiest thing, okay. Is introduction and selection, which is the easiest thing. The first thing to do is in case of selection, what we are going to do, in case of introduction, what we are going to solve. Sorry, there is an interchange in the serial wise. The first one, in case of introduction, it is the easiest and the fastest method. So we can introduce a resistant variety and introduce or acclimatize it in the new environment, okay. Or a parent which can cross, which you can make use it as a use to transfer the gene into the cultivated ones or the cultivar, okay. So these either two things can be done, primary introduction and secondary introductions, okay. Next thing is selection. Selection can be done in mass selection and two ways: mass selection, pure line selection in case of self-pollinated crop plants, mass selection and recurrent selection in case of cross-pollinated crop plants, and clonal selection in case of vegetatively propagated crop plants. So in case of selection, to avoid biotic and abiotic stresses, the main intention would be to broaden the genetic base, okay. So the best thing to do in case of cross-pollinated crop plant is to select or to do the recurrent selections, okay. And of course, in case of mass selection, when we are doing mass selection, we use natural selection, okay, natural cross-pollination and natural selection. So in this case, we are utilizing somehow the progenetic base of the naturally occurring mechanism, okay, natural selection. So in this way we can enhance the progenetic base, which reduces the amount of susceptibility to disease and insects, okay. Next thing is hybridization. Hybridization means not only formation of hybrid varieties, okay, although if we cross between two parents, diverse parents, and when we have a F1, so if we release the F1, it is called as hybrid varieties. But then there are other methods also: hybridization followed by selections, okay, we have already learned about it. So when the hybridization is done, the the transfer of genes from one parent to another can be done by backcross method, with the easiest method, and it can also be done with pedigree method. So when it comes to transfer of multiple genes, okay, so pedigree method and backcross method is used. Why multiple genes? Sometimes the disease resistance is not only controlled by a major gene, okay, sometimes it is controlled by two genes, sometimes it is controlled by more than two genes, okay. And most of the time it is, although it is controlled by an oligogenic genes, so in case of pedigree method, it is a method which we can use to transfer resistance for resistance from two different parents, resistant gene from two different parents, or we can also transfer genes from one of the parents and agronomic characters from other parent, okay. And we have already learned about gene pyramiding, which is introduction of more than two different genes from two different parents, okay, using several parents, okay. Using several parents, we are going to introduce several genes into a single host, okay. So in this process, it can be achievable with the backcross breeding method, step-by-step backcross breeding method. In case of step-by-step backcross breeding method, we have to do backcross breeding method every generations to develop each and every genes from several parents. It's a wild type collection from land races. Okay, genes from several other parents into a single host in order to make it a durable and sustainable variety, which can lead to horizontal resistance. Okay, and also the same mechanism can be achievable with pedigree method. Next thing is, if you don't have any variation in the environment, okay, so the most important thing is to utilize mutations. Okay, so if you don't have any resistance parent, resistance source, so the more you can go for mutation breeding. Okay, you can expose the cultivated variety or the wild type variety which is crossable with the cultivated one, and you can use it as a parent, or either you can release it as a variety. Okay, mutation breeding. Okay. Next thing is semi-clonal variation. So in case of semi-clonal variations, these can be obtained in two ways. First, uh, the plants is regenerated from cultured cells, or their progenies are subjected to disease stage, and the resistant plant is isolated. And that means when the plant, when the cell is cultured, it will form a callus. Again, the propagating callus will be subjected to disease resistant test by providing stress. Okay, so screening, by providing screening of the disease, and resistant plant will be selected for as a disease resistant variety. Okay, and the next thing is, secondly, the cultured cell are selected for resistance to toxin or culture filtrate production by pathogen, and the plant originated from the selected cells. Okay, the next thing is to select the resistant plant type from the toxin or culture filtrate screening. Okay, so this is a different kind of screening process in the cell itself, cell level itself. Okay, and in most cases these plants are also resistant to disease in question. Okay, so this is also another mechanism of or this is also another method of insect or disease resistance in the at the cell level. Okay, so though in this case we require much time and at the same less time, and at the same time the the variety, the selected lines is, we have to harden the selected line, which is another process of tissue culture. Okay, so another process of plant breeding. Next thing is genetic engineering. Example, good example would be BT cotton. Okay, so ending again to BT cotton because it is the only transgenic crop plant available and grown in India legally. So in this case, we in genetic engineering we can transfer any gene uh, from any source to any host. Okay, so there is no barrier in transfer, and compatibility is not an issue here. So we can transfer genes from one uh, source like bacterium to a plant body. Okay, and try out for its expression. So this is the case. So in case of BT cotton also, the gene is the Cry1AC is transferred from bacteria bacterium Bacillus thuringiensis Cry1AC, and and it is transferred into what, the cotton host. Okay, so this is it. If you have any questions, I'll be eager to help. Thank you.